Hey guys, so today we're going to talk about the ovary. And first, I'm just going to go over some important structures that you guys are going to see in the ovary. And then we're going to actually look at the slide. The ovary has a lot of theory, but I will not be going over all of this theory. You guys are going to go kind of do that on your own, but I will go over the most important aspects. So first things first, what is the ovary and what does it do? This is an image that I found from Google, and I think it's perfect for you guys to kind of just have an outlook of what the ovary looks like, because it's a bit difficult in our other slides to actually see all the main structures of the ovary. The ovary is a parenchymal organ, and it has two very important functions. The first function is oogenesis. This is the production of eggs. The second function is production of ovarian hormones like estrogen and progesterone. Now, I will explain later how these two things are done individually, but for now, know that the function of eggs is oogenesis and production of ovarian hormones, estrogen, and progesterone. Now, it's a parenchymal organ, so it's composed of your cortex and your medulla. Your cortex is this outside layer, and this is actually going to make up the most of your ovary. And there are two structures that you guys are going to see in your cortex. You're going to see follicles, which again, I will explain what they are in a second. And you're also going to see the stroma, which is very special. As it's a parenchymal organ, you have your cortex and you have your medulla. Your medulla is not going to be that important. Just know that it contains the vascular bed, the vegetative nerves, etc. Another thing that's very important about the ovary is lined up by simple cuboidal epithelium, which I'm unable to show right now, but I will show in the slide later. And under the epithelium, remember, under the epithelium, you have what's called tunica albuginea, which is a capsule, and it's composed of dense connective tissue. So, parenchymal organ, covered by simple cuboidal epithelium. Under the epithelium, you're going to have these dense connective tissue, which is a capsule that's protecting the ovary. And then you're going to have your cortex, which contains your follicles. And then you're going to have your medulla, which contains your vessels. Okay. Now, I mentioned that there is a stroma, and the stroma is very important. So, the stroma is called spinocellular. This is a very special form of loose connective tissue. So when they ask you about the stroma of the ovary, you say the stroma is loose connective tissue, but this is a special form of loose connective tissue. This loose connective tissue, which is called spinocellular connective tissue, is found in two places. You guys are going to see it in the ovary, and you guys are going to see it in the endometrium of the uterus. Well, why is it important? The ovary contains follicles that are going to be growing, right? They're going to be changing in size. And the ovary needs to be able to accommodate for that. By it having a stroma that's highly plastic, it's spinocellular, it allows for the changes in volume of these different structures. So it will grow when the ovarian follicles grow, or it will extend when they're growing, and then it would also shrink, which I will show you guys later why and when it shrinks. But basically, this is why spinocellular is very important, because it allows the changes in volume of the different structures that are inside of the ovary. What is inside the spinocellular CT? Inside the spinocellular CT, you will actually see fibroblasts. And these fibroblasts, they differentiate into very important cells. These fibroblasts that are here, this is the ovary. This is the spinocellular connective tissue. The fibroblasts which are here are going to differentiate into theca interna cells. These theca interna cells are going to be surrounding follicles, and they're going to secrete androstenedione. And the follicular cells, which you guys will then see what they are then going to turn this into estrogen. So remember when we were talking about the function of the ovary, we said production of hormones, estrogen and progesterone, and oogenesis. I will get into oogenesis in a second, but just know that we have the theca interna cells, which come from fibroblasts from the spinal cellular connective tissue. and they secrete androstenedione, which then turns into estrogen by the follicular cells. Now, let's go to the other function of the ovary, oogenesis. I'm not going to go into detail about this. You guys need to know this well on your own. But know that we have prenatal oogenesis and postnatal oogenesis. I think some of you might know the saying or just this knowledge that girls are born, newborn babies, yeah? Girls are born with all the eggs they're ever going to make. We do not make eggs after we're born. We are born with all the eggs we're going to have. These eggs are going to be stored in follicles, okay? Eggs are very sensitive structures. They're very sensitive. They need to be protected. They are going to be stored in these follicles, and these follicles are going to not only protect it, but they're also going to nourish the egg. They're going to allow it to grow. They're going to support the egg through production of hormones, such as estrogen. They also allow with communication, they also communicate or send signals to the egg, because imagine, right, you guys know that the egg is going to grow in the ovary, and then once it's grown enough and ready for ovulation, it's going to leave the ovary, it's going to travel through the fallopian tube and then go to the uterus. Sometimes the egg will grow so much, but the body is not ready yet. The uterus is not ready yet. Nothing is ready yet to accommodate the egg. The follicular cells, or the follicle which is surrounding the egg, is going to tell the egg to stop growing. It's going to keep it in what's called a meiotic arrest until everything else is ready to receive the egg, or until the egg is ready to leave the ovary. So these follicles are very important. I repeat, you have your egg, and your egg is surrounded by follicles. We, or women, are born with all the follicles, or all the eggs that they're ever going to have, and these are in primordial follicles. So this is what newborn baby girls have, and it stops there. Once you enter the fertile period, once girls enter puberty, then these primordial follicles are going to grow into a primary follicle, secondary follicle, Graafian follicle, and then ovulation is going to occur. Now, what triggers this, right? At what point do you go from primordial follicles to the growth of these follicles? Puberty. And what triggers that? Hypothalamus. You're going to have the release of gonadotropic releasing hormone, which is going to go to your anterior pituitary. Your anterior pituitary is then going to release this hormone called FSH, follicle stimulating hormone. So FSH is going to be released when a woman enters puberty, and FSH is going to act on the follicles and tell basically being like, hey, it's time to grow, or it's time to start activating, if that makes sense, to allow the egg to grow and then allow ovulation to occur. So what does the FSH do? The FSH is going to cause the follicular cells to grow and proliferate. So I'm going to go into a bit more detail so you guys can see the structure of these follicles and what I mean by that. So this is how a primordial follicle looks like. It has three basic structures. You have the oocyte, you have the follicular cell, and then you have a basal lamina. So these follicular cells are under the influence of FSH. These follicular cells are the ones that are going to be nourishing the oocytes. They're going to be protecting it, and they're going to be the one that are communicating to the oocyte. If it grows too fast, they're going to tell it to stop growing. And these follicular cells are also going to be producing estrogen. Again, remember, we have the theca interna cells, which secrete androstenedione, and these theca interna cells are right next to the follicle. So imagine that there are theca interna cells right here. They release androstenedione, and they release it into the follicle, and then the follicle is going to make estrogen. So FSH is going to cause the development of these follicular cells. These follicular cells are then going to make estrogen from the androstenedione that the theca internal cells provide to them, right? Why is this estrogen important? This estrogen is important because the estrogen, it creates a loop. Follicular cells are going to make estrogen, And the estrogen is then also going to continue allowing the follicular cells to grow. Another thing that the estrogen is going to do, have do you guys ever wonder why, like, in general, women don't give birth to like 6, 7, 8 babies? It's because we don't want, like, we do not have too many follicles growing at the same time. Usually, we want maybe 1, 2, 3 follicles to be growing, right? And out of those 3 follicles, in a wonderful scenario, 2 of the eggs are going to leave for ovulation and implant in the uterus, but usually only 1 egg is going to leave and implant into the uterus. We do not want too many follicles growing. So the estrogen that the follicular cells are making are going to go back to your brain and tell your brain, hey, FSH, you can stop, being secreted. We have enough follicles growing already. So again, we have your primordial follicle, oocyte, follicular cells, basal lamina. These are squamous follicular cells, and you're not going to see this. I tell you guys right now, you guys are not going to see this because these are more for the prenatal period and they tend to decrease. What you guys can see are primary follicles. You guys are going to see the exact same structures. You're going to see an oocyte. You're going to see one layer of columnar follicular cells now, and you're also going to see the structure known as zona pellucida. So this is the basic structure of a follicle. As it's growing, you're now going to have a primary multilaminar growing follicle. This is the exact same thing as this, but now you have a lot more follicular cells. Now you just have a lot more follicular cells. What does this mean? This means that you also have a lot more estrogen being produced. And the estrogen being produced is causing the follicular cells to grow even more, and at the same time telling the brain, hey, like, there's a lot of estrogen being produced right now. Do not secrete any more FSH. We do not want more follicles growing. We already have this follicle growing. We do not need more follicles growing. This is why estrogen is very essential. And estrogen is also preparing the uterus for the egg that's going to be released. And here you have the theca interna cells, which again, androstenedion, which is going to be turned into estrogen here. And the estrogen, you guys need to know this. I keep repeating it, but you guys need to know this. The estrogen is going to prepare the uterus, and it's also telling the brain to not secrete more FSH. We don't need more follicles. We already have this follicle. Or how many ever follicles we might have, 2 or 3. And so these were your primary follicles. In your primary follicles, you can see the oocyte is in the center, and then you have the follicular cells, and you have your zona pellucida, and here is your basal lamina. In your secondary follicle, there is a difference. You have these white spaces, which is known as antrum. This antrum is a fluid that has a lot of proteins and growth factors and stuff for the oocytes to grow and mature, etc. You'll also see that the oocyte is no longer in the middle. It's kind of deviated to the side. These are some examples of what secondary follicles are going to look like. You'll see these white spaces, which is the antrum. You'll see the oocyte. Here you're going to have your zona pellucida. Your follicular cells are here, making your estrogen. Here you're going to have your theca interna cells, making your androstenedion. Again, they came from the spinocellular CT, etc. You are not going to look at this and confuse it with this. Okay, here you see one layer of follicular cells. Here you see multiple layers of follicular cells, and you don't see any white spaces. There's no way that this is going to be secondary. While here you see this huge white space, which is the antrum, and you know that this is going to be a secondary follicle. You're not going to confuse this with a primary follicle. Now we have one more follicle to show you, and again, all these follicles, they are going to be in the ovary, like in different stages. Okay, you can see a primary follicle, and then you will also see a secondary follicle, and you'll see maybe, some primordial follicles. This is why the spinocellular connective tissue is important, because you're going to have these follicles which are growing, and the ovary needs to be able to accommodate it. So again, we went through our primordial, our primary follicle. Our primary has two. You have the unilaminar and you have the multilaminar. We went through our secondary follicle, which has the antrum. And now we have our tertiary follicle. This is our big follicle, okay? And they are called Graafian follicles. I mean, look at the difference between this and this. What is the major thing that you guys notice? The major thing that you guys notice is that the antrum is no longer a small thing. It is now taking up particularly the majority of the follicle. And this is basically a sign that we're almost there, okay? The egg is pretty much mature. It's like ready to leave the follicle, to go through the oviduct, and then go to the uterus. Graafian follicle, you are going to see an antrum. That is the number one thing you're going to notice. You're going to look at this huge white space. You know this is a Graafian follicle. It's also going to be much bigger than the other follicles. You're also going to see the oocytes, and there are two new structures that you're going to see inside the follicle. The follicular cells are all going to be moving to the side, right, to make space for the antrum, but they're also going to still be surrounding the oocytes. And so it creates this kind of crownish structure around the oocytes, and this is known as the corona radiata. And another thing is the oocytes is still going to be connected to these like follicular cells through a stalk, which you can't really see here, but you can see here. This stalk, which is still made up of the follicular cells, it's called cumulus ophorus. When you see the cumulus ophorus, you know that like any second now, the egg is going to be ready to leave the follicle and go through the oviduct to the uterus. Okay, so really quickly, guys, once again, babies, right, are born with all of their eggs. They're born with all of the follicles they're ever going to have. And these follicles are going to be in a stage of primordial follicles. Okay, they're not going to grow. They're going to stay like this until the baby or the young girl reaches puberty. Once they reach puberty, you're going to have gonadotropic releasing hormone acting on the anterior pituitary, and then suddenly you're going to have a release of FSH. FSH is going to act on the follicles and cause these follicular cells to start proliferating, to start growing. The follicular cells are going to start growing, and you're going to have the theca interna cells, which will be secreting the androstendion into the follicular cells, and now the androstendion is going to be converted to estrogen. It is not the FSH that's going to be keeping the follicular cells growing. You know, we started with one layer to multilaminar to even more follicles. This is not FSH, this is estrogen, because once those follicular cells are stimulated by FSH once, they start producing estrogen. And we don't want so many follicles being stimulated at the same time, right? Because we don't want 3-4 eggs to leave the ovary and then the woman is pregnant, you know, with that many kids. So the estrogen that the follicles are going to be making is important because it's going to be telling the brain to stop releasing FSH. Stop releasing FSH. You know, like FSH activated 1, 2, 3 follicles. These follicles are active, they're growing, and they're making estrogen, and the estrogen is telling FSH, you don't need to be released anymore. We have enough follicles being activated. And at the same time, the estrogen that they're releasing is going to be preparing the uterus. So they're growing and growing and growing to the point where they've reached the Graafian follicle. And by this point, you have so much estrogen, guys, so much estrogen that there's actually a switch. The estrogen is going to go from providing the negative feedback, right? Instead of telling the brain, hey, there's so much like of estrogen, stop releasing FSH, that instead the switch is going to be the estrogen telling the brain to release something. And this something is called LH. This is called luteinizing hormone. So when you are at the point of the Graafian follicle, there's so much estrogen being released by the follicular cells that the brain releases luteinizing hormone. It stops FSH or severely decreases FSH and is now releasing luteinizing hormone. What does this luteinizing hormone do to this Graafian follicle? It destroys it. So the luteinizing hormone basically is the trigger for ovulation. Once that luteinizing hormone occurs or spikes, this oocyte is going to completely rupture through this membrane. It's going to completely rupture through the follicle. It's going to travel to the oviduct, to the uterus, and now you can completely forget it. Now we have an empty Graafian follicle with no oocyte. What is happening to this Graafian follicle? Well, we know that we're no longer under the control of FSH. Now we're under the control of LH. LH is called luteinizing hormone. Focus on the word lutein, because now these follicular cells, which are also called granulosa cells, are going to be converted into granulosa lutein cells. The theca interna cells are going to be converted into what we call theca lutein cells. And essentially, the luteinizing hormone is going to destroy this Graafian follicle and form another structure called corpus luteum. So guys, corpus luteum are Graafian follicles that were destroyed under the influence of luteinizing hormone. You are going to see this structure only after ovulation has occurred. The oocyte has left the Graafian follicle, which was triggered by luteinizing hormone, which was triggered by all of that estrogen, which was triggered by the Graafian follicle, and now we have the structure, which is the corpus luteum. And there are two types. As you guys know, when there's ovulation and the egg reaches the uterus, there are two things that can happen. You either have fertilization or you don't, okay? This doesn't happen instantly. The corpus luteum is going to be secreting a couple hormones, or two hormones mainly. We have our granulosa lutein cells, right? Which were previously our follicular cells. They're going to be secreting progesterone and some estrogen because you still have some FSH stimulation. While the theca lutein cells, remember guys, when you guys see the corpus luteum, do not say theca interna, do not say just granulosa. It's granulosa lutein and theca lutein. Granulosa lutein will be secreting progesterone and estrogen, and the theca lutein are going to be secreting only progesterone. So we know why estrogen is important. We spoke about estrogen, providing that negative feedback loop so we don't have more follicles being stimulated and also preparing the uterus. But now we have now we have progesterone being released. Why is this progesterone important? Well, our bodies, or sorry, I keep saying our, the body is always going to operate under the assumption that fertilization will occur. So all of this is preparing the body for fertilization, okay? So the corpus luteum is going to be secreting progesterone. Progesterone is going to prepare the uterus. It's going to form the perfect environment for the egg to implant on the uterus. That's what progesterone is for. And it's still going to be, protecting the egg and just providing the best environment for the egg. When the body realizes that no fertilization occurred, we have what's known as the corpus luteum of menstruation, and it's going to regress in 2 weeks because it's going to be like, why am I still releasing progesterone when obviously there's no fertilization? But when there is fertilization, the corpus luteum is going to continue secreting progesterone until the 3rd month. Why? Because on the 3rd month, the placenta has formed, and the placenta will take over the corpus luteum. It's very important that if fertilization occurs, that you still have secretion of progesterone, because the progesterone is going to be maintaining that environment for the baby. So first, you have the we have progesterone being secreted until 2 weeks. When it realizes there has been no fertilization, it completely degrades. However, if the body realizes that there has been fertilization, you have HCG, which is another hormone being released, and the corpus luteum will continue secreting the progesterone because, again, we want to maintain that perfect environment for the egg until the 3rd month, which is when the placenta is formed, and then the placenta will be taking over, secreting all these hormones to protect the egg, and then this corpus luteum can degenerate. What does the corpus luteum degenerate into? The last structure, which is called corpus albicans. This is basically just fibrotic scar tissue of the previous corpus luteum. So now I'm just going to quickly show you guys the structures. So we have this first slide, which is of the ovary. Guys, there's literally not a single slide that looks like this. These huge empty structures, I did histology 4 years ago, but 4 years ago, we were told that these were mature Graafian follicles, okay? This is how big the Graafian follicles can get. Once again, spinal cellular connective tissue is very important because look at how big these Graafian follicles can get to. They need to allow the ovary to accommodate to these huge follicles. Yeah. So it was actually pretty difficult for me to find some other smaller follicles in this slide. I don't think it's a very well cut slide. The good thing is that for your slide test, you guys will not have to go looking for the follicle. The professor will find it for you and then just ask you what it is. But for example, I can see this follicle here, and I'm looking at the follicle and I see that we definitely know that it's not primordial, it is not primary unilateral. We see multiple follicular cells. So it's either going to be primary multilaminar or a secondary. I see some white spaces, so I would say it's secondary, but for your credit test, if you're able to just quickly point out the difference between how a primordial looks like, a primary looks like, multilaminar primary looks like versus a secondary, then you will be fine. I think if you say primary, multilaminar, or secondary, they will accept because it could be one or the other. Again, I'm not gonna be spending too much time, but it's really difficult to find sometimes, like when you see structures like this, it could be due to the cutting that you're unable to see the oocytes. But again, yeah, I believe it's too small to be a Graafian follicle. Usually when you see like a follicle without an oocyte, you know it's a Graafian follicle, but that seemed to be too small. Again, yeah, it's just a bit difficult for me to find, but some follicles, but anyways. So this was the ovary. You zoom out, you see the Graafian follicles. They will then somehow find a follicle for you, and you just let them know like whether it's a primary, secondary, and why. And you guys will be able to tell it apart because they do not look alike at all. Again, this is giving me very much. I cannot see the oocyte, but this is not a Graafian follicle. Sometimes it has to do with the cutting, or for whatever reason you can't see the oocyte properly. But you see the multi layers of the follicular cells. You can see the basal lamina. You do not see the oocytes, but essentially this would 100% be a follicle, probably a multilaminar primary follicle. Now, the second slide is the one where you guys can see the corpus luteum. Guys, the corpus luteum is huge, right? It's a Graafian follicle that was degenerated. It has to be big, and you see foldings. You have a lot of enfoldings. The Graafian follicle has completely folded apart. You see your granulosa, lutein, and theca lutein cells. Do not forget the lutein parts. Okay, perfect. Here we have a follicle, and this follicle is, we see the oocyte, we see the zona pellucida, we see the follicular cells, and then we see this white space. This is the antrum. This is definitely a secondary follicle. A Graafian follicle would be much bigger, and the antrum would be taking up the majority of the follicle cell. We see here a lot of vessels. They're going to ask you, you're just going to say, we're looking at like the medulla with the ovarian artery and vein, from the mesothelium for supply of straight cortical arteries into the cortex, yada, 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 yada. You guys are going to get that from your theory. But yes, this is, again, I cannot necessarily find more ovaries, but just memorize those images, memorize the structure. They're really not difficult to tell apart primary versus secondary versus tertiary, and just know that this, anytime you see you know that this is an ovary because you're going to see follicles. Anytime you see the follicles and then you see some huge enfolding structure, this is the corpus luteum. And this is it. Okay, guys, so now we are looking at the oviduct. The oviduct is the uterine tube, also known as the fallopian tube. This is basically where the egg is going to be traveling. The egg is going to travel at the lumen. There's nothing major about this slide, guys. You are going to see these anastomosing folds and a lumen in the middle, and this is going to be really your main diagnostical feature. It looks like, I don't know, a leaf or a tree. Your tunica mucosa, you're going to see, again, branched long anastomosing folds, and they're occupying the lumen. Simple columnar epithelium. You're then going to have your tunica muscularis externa, which is two sublayers of smooth muscle tissue, and you have your tunica serosa, which is your mesosalpinx. Your other slide, again, the main diagnostic feature is really just looking at these anastomosing folds that are covering a lumen. The lumen, or the white part, is where the egg is going to be traveling. In case, yeah, so this is, again, you're not really going to see any other interesting thing. Just the lumen, the anastomosing folds, the tunica muscularis externa, and know that the tunica serosa is called the mesosalpinx. Now, you guys have two slides for the uterus. You have one slide which is called the proliferative slide, and you have the secretory slide. And they are called after two very important phases that are happening in the uterus. Let's start with the first phase, which is known as the proliferative phase. So guys, the menstrual phase, or sorry, the menstrual cycle, you guys know starts with bleeding, okay? You know that once a month, women are going to bleed. And you know that this happens when there is no fertilization, when a woman undergoes ovulation, but there's no fertilization. That bleeding, a lot of people assume is just the egg, right? The egg will completely undergo necrosis, the oocyte will undergo necrosis, and then it's gonna turn into blood, etc. But it's not just the egg. It's the egg and part of the uterus. What do I mean by that? The uterus is composed of 3 layers. You have the innermost layer. This is the inside of the uterus. It's called endometrium. You have the middle layer, which is called myometrium. It has 3 very thick, smooth muscle layers. And you have the outer part, which, depending on where you are in the uterus, it could be perimetrium or parametrium. And then you guys can see on your notes which is what, where, etc. So during the menstrual cycle, when the egg is implanted in the uterus and there is no ovulation, no fertilization, you know that there's going to be necrosis. There's going to be bleeding. I'm telling you right now, that bleeding is not just the egg. It's the egg that undergoes necrosis. But you guys know that the egg is implanted into the uterus, right? It's implanted into the endometrium, the innermost part of the uterus. And when the egg dies, that endometrial part dies with it. But that endometrial part is very, very important, right? Because when the egg is implanted, the endometrium has glands, and these glands are going to be secreting all the nutrients that are needed for the egg. They're going to be creating the perfect environment for the egg. So when this endometrium part dies with the egg, when there's no fertilization, the uterus doesn't just stay like that, okay? The endometrial part is rebuilt again. It's remade. And this is where your 2 slides differ. Your first slide, guys, is your proliferative phase. Your proliferative phase is when the endometrium is being regenerated. It's happening right after menstruation. You have basically no endometrium. You only have a little bit of some endometrium. How do you guys tell the endometrium apart from the other layers? The endometrium contains glands. And when it's being regenerated, when it's being rebuilt, these glands are very straight and narrow because they're not activated yet. They're not secreting anything yet. You have your early proliferative phase, which is you have the straight tubular glands, and then you have your late proliferative phase. And in your late proliferative phase, the glands are starting to elongate and the endometrium is going to start becoming a little bit thick. See, now it's very thin. This is early proliferative phase, but later it's going to become much more coiled and it's going to become much more thick. This entire thing, or it's going to grow a lot more. Now, what is causing this growth? It's very important that you guys know this. everything that is happening in the uterus, the destruction of the endometrium and the necrosis of the egg and the endometrium, and then later on the regeneration of the endometrium. All of this is a process. And there's a very similar process that's occurring in another place that we spoke about, and that is in the ovary. So in the ovary, we spoke about how we have the follicles, right? The follicles are making estrogen. The estrogen is causing the follicles to grow. Remember how I told you guys that the estrogen has another important job? Or what does estrogen do? We said estrogen provides the negative feedback loop to the brain, telling the brain not to secrete more FSH, so we don't have a lot more follicles growing. We said that estrogen also helps the follicular cells in that particular follicle grow or proliferate. And we said that estrogen also prepares the uterus. Remember? We said that estrogen prepares the uterus, causes changes in the uterus to receive the egg. These are the changes that I'm talking about. So when you guys get this slide, when you guys look and see a very thin endometrium, you zoom in, you see this very thin, straight glands. They ask you, what is this phase? You guys are going to say, this is the proliferative phase. They're going to say, okay, what else can it be called? You guys are going to say it can also be called the estrogenic phase or the follicular phase. Because when the follicles are growing in the ovary and when they're making estrogen, the estrogen is causing this. So this is happening at the same time that the follicles are growing. This is happening at the same time that you have your primary, secondary, tertiary Graafian follicles. They're all happening at the same time. Then what happens? What happens when you get your Graafian follicle? You have ovulation. What triggered that ovulation? The luteinizing hormone. When you had too much estrogen, this caused a positive feedback, and you have luteinizing hormone released. That luteinizing hormone caused what? It caused the ovulation, it caused the egg to leave the Graafian follicle, and it caused the degeneration of the Graafian follicle into what? The corpus luteum. What does the corpus luteum secrete? It also secretes a hormone called progesterone, right? And the same way that estrogen was very important for the proliferation of the uterus, of the endometrium of the uterus, the progesterone is, well, I don't know if the estrogen, I don't know if I made a mistake. The estrogen is important for the proliferation of the endometrium. The progesterone is also important. So the follicles and the estrogen phase is important for this, but the corpus luteum and the progesterone phase is also important. What do? Well, they basically tell the uterus that the egg is on its way and that these glands need to basically activate and prepare to receive the egg. And so when you have your second slide, which is your secretory phase, you can also call your secretory phase your, this is not the one I wanted. You can also call your secretory phase your progestational phase or your luteal phase. And look at the difference. Look at these glands compared to these glands. You see, when the endometrium is proliferating, when it's under the effect of estrogen, you have straight, narrow glands. But when it's in the progesterone, because guys, the corpus luteum is created after ovulation. So the egg has officially left the Graafian follicle. The egg has left the ovary. That means that any time now, the egg is going to be implanting in the uterus. So the uterus needs to be ready, right? It needs to be ready to receive the egg. So of course, when there's corpus luteum, there's been ovulation. And corpus luteum is secreting progesterone. So when the endometrium is like this, ready to receive the egg, you know that this is under the effect of progesterone. This is in the progesterone phase, because the progesterone phase is when the endometrium is ready to receive the egg. And these glands, why is it called secretory phase? It's called secretory phase because the glands are going to start secreting all the nutrients needed to keep the egg. And so not only do you see these large coiled glands, look at that thickness of the endometrium. It's almost pretty much half of the size of this complete slide. And it's so much thicker that you can even see two layers inside of the endometrium. Look at this. Look at the difference in shape between these glands and these glands here. Okay, here we see the big glands with large like lumen, and here we see straight narrow glands, almost like the ones in the proliferative phase or estrogen phase. That's because the actual endometrium has two more layers within it. This is the functional layer, and this is the basal layer. So now that we're in the progestional phase, we know that the egg is here. It's on its way and it's arrived and it's here. And there was no fertilization. We said that when there's no fertilization, we have necrosis of not just the egg, but the endometrium. Well, it's the functional layer of the endometrium that dies, that bleeds out. The basal layer stays. And when the entire cycle repeats again, when estrogen is starting to be made by the follicular cells, the estrogen is going to cause proliferation of the basal layer. The basal layer that stayed after menstruation is how the estrogen is then going to rebuild the endometrial layer of the uterus. So it's important to understand proliferative phase, you have proliferation of the endometrium or rebuilding the endometrium after it's died, after the menstruation. And the proliferative phase is also known as the follicular phase or estrogenic phase because you have a lot of estrogen being made by the follicular cells, and it's the estrogen that's causing the growth, that's causing the proliferation. Once you have the ovulation and you have the corpus luteum, and the corpus luteum is now secreting progesterone, this is what's going to happen. You're going to have these large coiled glands, and not only is this, and they're secreting, right? They're providing the nutrients for the egg, and not only is this called the secretory phase, it's also called the progestational phase or luteal phase. Okay, and our last slide is going to be not a very difficult slide. It's the vagina. And the vagina, it's very simple, guys. You have your tunica mucosa, where you're gonna have your epithelium. I don't even have to zoom in to know that this is going to be stratified squamous non-keratinized. Okay, but when I do zoom in, one thing that I notice is that there are a lot of light cells. What are these light cells? This is basically just your epithelium that is accumulated with glycogen. Okay, why is it accumulated with glycogen? Glycogen protects the vagina from pathogens. You know how when we had something happen, we had this entire process happening in the ovary, at the same time we had some changes happening in the uterus. Well, at the same time, you also have changes happening in the vagina. So when we have the estrogen phase in the uterus, which causes the proliferative phase when we have the estrogen phase in the ovary, causing the proliferative phase in the uterus, you also have the proliferative phase here in the vagina. You have proliferation of this epithelium. It's going to become very thick, full of glycogen, okay, protecting the vagina from pathogens. Once you have the progesterone phase in the ovaries or the secretory phase in the uterus, you have desquamation of this epithelium. It's going to become much thinner, not thicker. So just be aware that there are changes that are happening in the epithelium of the vagina. You then have your tunica muscularis externa, which are two layers of smooth muscle, and then you have your tunica adventitia, which has all these thick bundles of elastic fiber. And this is really it for the vagina. There's nothing more. There may be some details that you need to know, but we don't really have much time. But you're going to see a stratified squamous non-keratinized epithelium with very light stained cells, and then you're going to see tunica muscularis externa, tunica adventitia, and nothing else. That there's no other differential diagnosis that's going to tell you that you're in the vagina. You basically just think, well, I don't see kind of anything else that might indicate whatever, so this is the vagina.
All right, hello guys. Welcome to our seminar for the male reproductive system. So I'll be talking about the spermatic cord and the seminal vesicles. So let's begin. Uh, these are the two slides which are in the fingerland. Both of them are the same structure. It's the spermatic cord. Now let's look at some theory and then I'll explain you guys the slide. So when we talk about the spermatic cord, it is a really important structure that connects, the testes to the rest of the body. Now, it stretches from the deep inguinal ring and then it goes through the inguinal canal and then it ends in the scrotum. Now, inside it, there are several components and these are, bundled together in the connective tissue. The most important one, which you'll see on the slide, is called the ductus deferens. And alongside it, there are, the testicular artery as well as the artery to the ductus deferens, which bring the blood supply of the spermatic cord essentially. When we talk about the venous supply is done by the pampiniform plexus, which helps in, the temperature regulation of the testes as well. And when we talk about a specific property of this plexus, it has a tunica media, which has a circular smooth muscle, and the tunica adventitia, which has a longitudinal smooth muscle. And another characteristic property of the cord is that it has the cremaster muscle. It is a skeletal muscle, also the striated muscle. What it does is that it, helps raise and lower the testes in response to temperature or any kind of other stimulation. And finally, we also have the lymphatic vessels as well as the nerve plexuses. These help in, the immune function as well as the innervation of the spermatic cord. Now, as you can see, these are the pictures that I have attached, but we'll look at the slide here. Uh, so first of all, on this part, you can see what are these. This is just looks like the striated or the skeletal muscle, because the nucleus is flattened and it is present at the periphery. So this is the cremaster muscle. All right. As you can see, the nucleus is present at the periphery. So this is the cremaster muscle, and this is one of the most important parts during the differential diagnosis. Then we have the uh, ductus deferens, which is really prominent in this slide as well as the other slides. So cremaster muscle and the ductus deferens would be your differential diagnosis for this slide. So when we talk about the theory about the ductus deferens, I'll explain that in the other slide, but we'll look at the features right now. It's not a problem. So when we talk about the tunica mucosa, it is lined by, the pseudostratified columnar epithelium. How do you know it's columnar? Because the cells are, elongated at the bottom, at the top. And you can see these hair-like strands present at the top of the epithelium. These are called sterocelia. So that's another property. And then we have the tunica muscularis, which is made up of two layers. It has the longitudinal muscle layer as well as a circular muscle layer. Now, on this part, this is not labeled here, but on the outer part, this is the tunica adventitia, which is made up of, the loose connective tissue, vessels, and nerves. Another characteristic feature is the veins. These are the veins which form the pampiniform plexus, as I explained their characteristics. I'm sorry, this is the artery, this is the vein, yes. Yes, these are the thick-walled veins which form the pampiniform plexus. Now, the difference between, the veins and the artery can be confusing, just like how I confused it now, but veins will have a quite larger lumen compared to the arteries, and you can see the thickness of the tunica media. It is much more thicker in the artery compared to the vein. Now, as I said before, the thick-walled veins, they have smooth muscle in both the tunica media as well as tunica adventitia. And in adventitia, it is longitudinal smooth muscle, and in tunica media, it is a circular smooth muscle. When we look at the other slide, it's essentially the same. It has the same features. As you can see, this is the cremaster muscle because it has the nuclei located at the periphery. Uh, yeah, that's about it. And then this is the ductus deferens. Again, the same properties, tunica mucosa, tunica muscularis, tunica adventitia, and then this would be the thick-walled veins. So when we talk about the ductus deferens, as I said, it has a star-shaped lumen and three layers: tunica mucosa, tunica muscularis, tunica adventitia. When we talk about the tunica mucosa, it has the pseudostratified columnar epithelium with sterocelia and folded lamina propria mucosa. And the tunica muscularis, it has three layers, inner longitudinal, middle circular, outer longitudinal as well. When we talk about the tunica adventitia, it has vessels, nerves, and it basically connects the ductus deferens to the spermatic cord. When we talk about the function of the ductus deferens is to, it provides a powerful contraction during ejaculation. So it pushes the sperm out of the ductus epididymis as well into the ejaculatory duct and then into the urethra. Now, moving on to the seminal vesicles. These are your two slides for seminal vesicle in figure land. When we talk about the seminal vesicles, they are actually, a part of a pair of coil-shaped glands that play a key role in the reproductive system. So they look like a bunch of tubes, but it is just really the same tube looped over and over and again. When we talk about structurally, their innermost layer is called, the tunica mucosa, which is superfolded. And this, what it does is, this folds basically help increase the surface area for the secretion of the seminal vesicle. And this lining is, mix of simple as well as pseudostratified columnar epithelium, depending on the thickness of these folds. When we talk about the middle layer, it is the tunica muscularis. It is made up of smooth muscles, circular on the inside and longitudinal on the outside. When we talk about the outermost part, that's the tunica adventitia. Again, loose connective tissue, contains blood vessels, lymphatics, nerves, and even sometimes it has fat cells. Now, one more important thing to remember is that because these tubes are so tightly coiled in the center, yeah, this layer, the tunica adventitia, can be, very thin, so it is really hard to see sometimes on some slides, okay? So there's that. And when we talk about some theory regarding, the function of the seminal vesicle, uh, first of all, they produce testosterone. No, the secretion is testosterone dependent, and the form of secretion is apocrine. When we talk about the secretion is slightly alkaline, which is super important because it helps neutralize the acidic environment of the vagina. So it basically makes the environment safer for the sperm. When we talk about the color of the secretion, it is yellowish color because of a pigment present called flavin, and the secretion is packed with useful nutrients. This includes fructose and citrate, which provides energy for the sperm. Then we have the proteins, which help nourish and activate them. Then we also have the fibrinogen, which causes the semen to coagulate after ejaculation. And then we have some prostaglandins, which help stimulate the female reproductive tract muscles as well. So overall, these glands, they set up the stage for the sperms to survive and move through the female reproductive system as well as succeed in fertilization. Now, when we look at the slide for the seminal vesicle, as you can see, it looks like coiled tubes. Uh, but this is the main structure which we're looking at. So this is the lumen of the seminal vesicle. And when we are talking about the epithelium, this small structure, as you can see, the upper part of these cells, they are circular. So this shows, the transition between the simple cuboidal to the pseudostratified columnar epithelium. And when we talk about different types of folds, there are two types of folds present. Here we can see the secondary fold, which again, as I said, they help increase the surface area for the secretion. And uh, this is a primary fold. So this is the primary fold, which then leads, and from this branches of the secondary fold. Okay, and underneath the mucosa, you have the lamina propria mucosa, again loose connective tissue, and then here the pink part in the lumen of the seminal vesicle. This shows the secretion, the apocrine secretion of the seminal vesicle, and the outer part, it shows the circular layer of smooth muscle. Uh, I don't think there's anything else in this slide. Sometimes in this slide, you can also see the ductus deferens. Again, this is the ductus deferens. These are the muscle layer, inner longitudinal, middle circular, and outer longitudinal layer. And then this is the tunica adventitia. And overall, combined, this is called a macrotubule network. Again, the other slide, it almost looks the same. Here you can see the ductus deferens here, the macrotubule, again, the secretion, the pink parts represents the secretion, primary fold, secondary fold. So, yep, that's about it from my side. Thank you.
I'll be talking about the testis, the epididymis, and the prostate. So this slide over here is of the testis and the epididymis. This part over here is made up of the head of the epididymis, and this part over here is the testis. So the testis is a parenchymatous organ, which means it has a stroma and a parenchyma. The stroma consists of a capsule which lines the testis. So this capsule over here is made up of dense connective tissue, and it is called as the tunica albuginea. This capsule sends septa into the testis to form approximately 250 incomplete lobules, which are called as testicular lobules. And this is where the stroma ends. Then we have the parenchyma. The parenchyma is made up of seminiferous tubules. So each of the testicular lobules has approximately 1 to 4 seminiferous tubules. And between the seminiferous tubules, over here, for example, we can see that there is some sort of a connective tissue. So this interstitial connective tissue consists of these very important cells, which are called as Leydig cells, and they produce the testosterone. The wall of each seminiferous tubule is made up of loose connective tissue and smooth muscle. So the seminiferous tubule consists of the special epithelium, which is called as the seminiferous epithelium. And when the males reach sexual maturity, the seminiferous epithelium starts getting composed of these Sertoli cells as well as the spermatogenic cells. So these Sertoli cells, they have a really pale nucleus. So this cell, for example, here, this would be a Sertoli cell, and let me see more. This one would be a Sertoli cell, and this one would be a Sertoli cell. So the function of these Sertoli cells is to form the blood testis barrier and also provide nutrition to the developing spermatogenic cells. So spermatogenic cells, there's approximately 5 stages of them. So the first one being the spermatogonia, and they are like the very first stage of the sexual gametes. So the spermatogonia, this one with a really dark nucleus over here, and this one would be the spermatogonia. They're located like near the lining over here, and not like near the lumen. They're located on this side. So um, these then undergo division to form primary spermatocytes. Primary spermatocytes are situated above the spermatogonia, and they are the largest cells. Their nuclei is has a characteristic fibrillary appearance, and they are usually present in the first meiotic division. So they will have 46 chromosomes and 2 chromatids. So this one over here would be a primary spermatocyte, and so would this one, because you can see there's a characteristic fibrillary appearance of the nucleus. These then further undergo division to form secondary spermatocytes, and they are usually not seen in the seminiferous epithelium because they are very short-lived, and they immediately enter the second meiotic division to give rise to the next cells in the lineage, which are called as spermatids. So the spermatids are very small, and they have a rounded nucleus which is also really dark. They're located right next to the lumen. So this would be a spermatid. So with this and so with this, so all the small shaped cells which you can see here, these ones are your spermatids. These spermatids further undergo a process which is called as spermiogenesis, which forms the mature spermatozoa. And these are seen like near the lumen, and they have kind of like a droplet like a shape. So this structure over here, this one would be a mature sperm. Or let me try to see if I can find other ones. Yeah, these ones, these tiny structures which don't have a circular nucleus, are just like kind of like a raindrop shape. So these ones are your mature spermatids. So just to summarize everything I just said, seminiferous tubules lined by loose connective tissue and smooth muscle. They have Sertoli cells with a really pale nucleus. So this one, this one, this one, and then they have spermatogonias, which are this one with a dark nucleus. Then these undergo division to form primary spermatocytes, which have a fibrillary nucleus. So this one would be a primary spermatocyte. So would this one be? These undergo further division to form secondary spermatocytes, which you can't really see because they're really short-lived. Then spermatids are formed, which is this one, very close to the lumen with a really small nucleus, which then undergoes spermiogenesis to form a mature sperm. These are the sperms. Now, when we move on to talk about the epididymis, the function of the epididymis is basically to store sperms and to carry them, to transport them basically. There's 2 parts of the epididymis. They're called ductula afferentis and the ductus epididymis. The ductula afferentis is the part which is closely attached to the testis, and it is made up of approximately 10 to 20 convoluted tubules. Um, It contains of the star-shaped luminal cells, and which are lined by either simple or pseudo-stratified epithelium. And the cells alternate between ciliated columnar cells and non-ciliated cuboidal cells. So the function of the non-ciliated cuboidal cells is to absorb the fluids which are produced by the seminiferous tubules, and the function of the columnar ciliated cells is to beat towards the ductus epididymis, which helps in the assist of spermatozoa basically. So when I zoom in over here, so the only thing which is important over here is like you need to know the epithelium, and that's honestly it for the ductula efferentis, because you can't really see much in the slide. Ductus epididymis, there is no slide on it, but I will just quickly give you a brief about it. So it is a single tube which is approximately 4 to 6 meters long, and it forms the body and the tail of the epididymis. Its lumen is lined by pseudostratified columnar secretory epithelium, and it consists of the sericilia. So basically, they are non-motile branched microvilli, and yeah, so basically the function of the ductus epididymis is to store the sperms and to transport them. Moving on to the slide of the prostate. So just some brief theory about the prostate. It is accessory genital gland which is located right under the urinary bladder, and it also consists of part of the male urethra, which is called as the prostatic part. So the external part of the prostate, which is over here, consists of a fibrous capsule, which is made up of dense connective tissue and a mixture of smooth muscle cells. You can see a bunch of vessels over here. So there's arteries, veins, and a lot of vegetative nerves, which are responsible for the innervation of the smooth muscle, which you can see there's a lot of it present in the capsule. Then right below the capsule, all of this area, which you can see like from the capsule, all of this area is going to be called your fibromuscular stroma, and it consists of a mixture of dense connective tissue and smooth muscle, which is like arranged very irregularly. So all of this is a fibromuscular stroma, and there are these special glands which are called as prostatic glands, and they are embedded into the fibromuscular stroma. So all of these glands, which you can see, all of these cloud-shaped glands, they are called as the prostatic glands, and their lumen has a very irregular shape. Um, the epithelium which lines them is either simple columnar epithelium to pseudostratified columnar epithelium. It kind of varies. And very important point to note about these glands is that they undergo apocrine secretion. That's why you will not find a very constant shape of the epithelium here, because some cells are already undergoing the apocrine extrusion. So yeah, the shape basically varies. Uh, that's it about the prostatic glands, and they secrete a bunch of things which I will talk about shortly. But there's one more important feature of the prostate, which is called as the prostatic concretions. This one is a prostatic concretion, and what it basically is that the substances which are produced by the prostate, they sometimes condense and they calcify to form the structure over here, and it is actually really harmless, but it increases with age. And yeah, that's it for the main slide and the differential diagnostics of the prostate. So let me talk about a bunch of theory which is really important. The prostate, like I said, is an apocrine gland, so its secretion is a liquid which is slightly acidic and with a pH of 6.5, and it consists of a bunch of important components. First one being the citric acid, and it also contains lipids for the nourishment of the spermatozoa. It consists of zinc, which is really important for forming the glycocalyx layer for the spermatozoa, which is essential for giving it nutrition and for transport, and also during fertilization. It also secretes something called as fibrinolysis, which is a proteolytic enzyme which helps in the decoagulation of the ejaculate, and prostaglandins and immunoglobulins are also secreted by the prostate. So that's it for the slide of the prostate.